my presentation is going to be a little bit different regarding the others we were presenting here. Why? Yeah, I would like to use this one.
And this is the laser print.
Yeah, okay. Because in this case, I'm not going to talk about me, what I'm doing regarding research with DNR Barcode, but as a person that was involved in a project developed by this university along with Peru. The name is DNA Barcoding to support the conservation of biodiversity, its sustainable use, and trade. When I found this slide, I said, what to do in this case? Well, I went to the iBOL systems and looked for whatever they have regarding Peru. So, the last week, these were the figures with more than 19,000 sequences. And you can see over there 115 institutions collaborating to reach at the present time those numbers. Yesterday, I went again, and the numbers increased a little bit. And you can see here so many institutions, and among them, we have 2 Peruvian institutions highlighted, an NGO, and a national university, San Marcos University. These 2 institutions are working a lot in ornithology, so most of the sequences they contributed with are regarding bird species. But I would like you to know this part in here. Most of these sequences come from GenBank. So this would give us a photograph of what's happening in Peru. Sure, we have good groups working in biodiversity, a good part with DNA too, but perhaps more of them are working towards evolution, phylogeny, this kind of science, well, part of the science. So when they're going to publish, they send the sequences to GenBank, and not necessarily are connected with a BOL system, So when we had the opportunity to work along with Guelph, developing this project, we had really the opportunity to gather everyone among us, to talk about this, and probably to go ahead as a nation, really, and contribute in this big opportunity. It wasn't easy. At the beginning, we had to work with around 40 people, 40 persons. It was mandated for the project, but we said, yeah, it could be. But if we do it this way, at the end of the project, it's going to be nothing, just a couple of institutions and a conversation among probably a couple of other institutions more. So we decided to stretch as much as possible. So at the end of the project, we talked with more than 300 people, around 70 institutions. So the idea was probably at the end to have a network. And actually we did, but nobody really was committed, especially with this, because most of these people, most of us, perhaps everyone, didn't represent exactly to our institutions as legal. For example, if I say, yeah, I'm going to sign this paper because we would like to do this to increase the knowledge of our species through the DNA barcode and all these things, it's really we would like to do, but I don't know if my, I don't know, my principal or for the minister, whatever, these kind of personalities could agree with this. And the other problem was that the minister, for example, agricultural, the environmental ministry, didn't want to sign anything because they say, okay, it's going to be okay. But what about Nagoya? What about many other things? Because Peru signed so many of these kind of papers that are going on around the world that it was very difficult. But anyway, at the end, we could say that we reached many groups that before this never was possible to do it. Because we could gather of these people, many other groups, for example, business groups and all the things, could be seated with us and say, okay, it's good. It would be one thing. The other one, for example, the transport minister or something like that, they say, oh yes, this is a good idea. We have so many problems with aviations, this problem of birds, the strikes and all the things, so yeah, we should work with this. I think this was the most important achievement we get in order to continue working on this. Anyway, so these two institutions are working, many other little ones. They are contributing to, also with iBOL, but really with a few samples. So in here, we're saying that really we are not a network. What we have is, we are not a formal network, but a willingness to work for it. The Natural History Museum of Natural History of San Marcos University is leading it at the present time. What we're doing is to push as hard as possible in order to gather more institutions to go ahead with these initiatives. When we get the invitation to be involved with this second phase, our first idea was, oh, we have to go again with the Minister of Environment and talk about this because we as people cannot sign anything because we're not really a network with papers and all the things. And if it is going to be a national thing, probably the good thing would be to have one of these ministers signing it. We went there, they were so happy, they said, okay, yeah, we're going to do it. If we are not going to pay, it will be so easy. We just sign it. But because this, I don't know. So at the present time, we don't have the amount signed, sorry, we're trying. But anyway, a couple of institutions were working really together. It was San Marcos, and the other one was SENASA. How competitive fundings, government fundings, and our wishful thinking to barcode as much as possible of the Peruvian megadiversity. With iBOL as a network or not. So after this Barcode project that we have with Guelph, appeared another opportunity for funding, because our government, exactly as Manuel mentioned for his country, they are opening funds, but always with something completely applied or with business. So one of these group of business went to us and said, really, it's very interesting. I found a good opportunity to use our tourist facilities and probably to apply one of the things over there. So at the end, they won. Leti Salinas is an ornithologist. She is the curator of our collections of ornithology in San Marcos University. And finally, we got a project and it's underway. Here I would like to read this brief description of goals, a collaborative project between a private company and academia in Tambopata, Peru, from 2015 to 2023, that includes the inventory and documentation of the biota or rainforest expedition, tourist reserves outside and within protected areas. For example, this one, Bawaja-Sonene and Tambopata, with the aim of contributing to the digital DNA barcode library and searching for new species for science. The number of target species and specimens of birds is 710 species and 2840 specimens, whereas our number of target Lepidoptera species is 2,000 with 800 species. It's working really nice, mainly for moths, because they use the light trap, and the tourists go ahead and look and see, oh, how nice, look at this, look at that. I'm going to pick this one. So the scientists around there, they say, okay, we're going to sequence the one. If this is a new species, it's going to be probably named with your name or something like that, and it's going like this. The part of birds is a little bit more difficult because it is not possible just to take tissue as easy as possible. So they're working with feathers and all kind of things like that. But it's very difficult because of the sensitive also. They're saying, oh no, you can't touch these animals and all this. But anyway, it's going ahead. And we have these institutions, sure, we're with the University of Guelph too, but I would like you to see this other. This is part of the government for the permits, and this is the worst thing, the terrible thing for us. I think that our projects in Peru, regarding exactly what we're talking about here, is because Leti Salinas is very persistent, and she goes and goes and goes. With every paper, they ask to go ahead with the permits. In this case, Leti really got some permits, but it is not possible to work in protected areas because we don't have the permits yet, and the time is passing. I love this part regarding SENASA. This is an institution from the government, sure, but since the beginning, their leaders were so open that it was possible to go ahead even within the project. Now they are going ahead with a particular project. In this case, we can say here our main interests are the arthropods that could be possible pests for crop of interest. We started our evaluation of possible pests in crops such as asparagus, quinoa, and Andean cedar. And here the numbers they have and they hope to have.
We're running.
Time?
Yes.
Okay. Finally, just to show you that there are some other groups that are going ahead, and finally, for example, this group get a fund for sequence at least 1,000 specimens, 1,000 species from our marine environments. We have so many difficulties. I think we have more difficulties than other things, but we are a mega diverse country, and we have the hope that we are going to get this. We are going probably not as much as you're doing because we have so many things against the initiative, but little by little, I think that we're going to get it. Thank you very much.